Okay, we are recording, just so you know, and the recording will be shared by Jacqueline through a newsletter later, and also posted to the AgBioData website. So thanks to everyone for joining us today for our monthly AgBioData general meeting and webinar. Great to see everyone on. Hope you're all doing well. We don't have any general announcements today. I think steering committee, please correct me if I'm wrong. So I was going to jump right away into the meat of it. So well, first I wanted to mention, so today is one of our general webinars. And this came about because now, as you all hopefully know, each of the AgBioData working groups should be volunteering 2 webinar discussions per year. And so this one came about from discussions in the data sharing working group that's led by Meg Staton and myself, Meg, sorry. So, and we were discussing in one of our working group meetings how even the somewhat seemingly trivial task of sharing just nucleotide and protein sequence data and metadata is still a challenge in terms of how different databases still decide to model and format that metadata. And that got us to thinking to ask the Alliance for Genome Resources that has been formed to help unify the main model organism databases funded by NIH to share their data and create a common platform. And so we reached out to Sierra Moxon, who is a developer within the Alliance to help us out there. And so she's going to give us a talk today on data management and sharing within the Alliance for Genome Resources. Briefly about Sierra, I had the pleasure of looking at her CV, and she's had a really great career of the intersection of biology and computer science, like I think a lot of people on this call have experienced as well. She had got her education in biology and computer science, then moved on to do developer work primarily in commercial settings initially, but then seemed to start in 2003, I think, Sierra, with the Zebrafish Information Network, and she's been working with ZFIN since then, doing all sorts of development, full stack, data munging, ETL, all those things, and also education and training as well. So she's been, looks like a linchpin of the ZFIN project. And she's also been working with the Alliance for Genome Resources as a software developer and data specifications lead since 2016. So she's well situated to give us an overview of how that's been going for the Alliance for Genome Resources. So Sierra, go ahead and take it away. Let me know if you have trouble sharing your slides, but hopefully that'll work out now.
Okay, thank you very much. Let me go ahead and share my screen here. And I'm going to put it into presenter view. So please let me know if the slides aren't advancing. Sometimes there's some magic here with the screen share on Zoom. But yeah, I'm Sierra. Thank you for that lovely introduction. I wanted to talk today about Alliance data management and how we've been evolving those best practices over the last 4 years now. And if you have any questions, please just speak up, yell into the microphone, Sierra, stop. I'm happy to answer them. I hope it's a good discussion rather than just me talking to you for an hour or so. So to sort of get started, just give you a little bit more of a brief introduction about the Alliance. The Alliance is really a consortium of 7 different knowledge bases, including SGD, the yeast genome database, worm-base, fly-base, ZFIN, RGD, the rat genome database, and MGI, the mouse genome information database, and also the Gene Ontology Consortium. And our mission is really to try to make information resources, both web-based and other, that facilitate the use of diverse model organisms in understanding human biology. And the substrate for this project was really 7 established organizations. We all had our own software engineering practices and websites and all sorts of things. So you can imagine challenges. What challenges? We don't have any challenges. I'm sure, as you understand in the AgBioData world, the consortium, that we all have similar data and we have a similar purpose, but we have very different software architectures and we have different styles of development, different data priorities, and really diverse user communities. And so our challenge is to bring all that together and really make a happy and wonderful user experience that can compare lots of different species together. So I just wanted to give you kind of a blank slide. This is where we started at the Alliance, and we grew from there quite quickly. We started with genes. They seem to have the most similar attributes that we could synchronize on. We quickly added some disease annotations, orthology. We added phenotype and expression data and all sorts of ontologies. And we've got interactions in there, and I'm just sort of looking at all our different nodes. This is a view into our Neo4j database. It has lots of nodes and relationships. And so what we like to say is we kind of got to a period of punctuated equilibrium. We have all sorts of base level objects here. We have genes, alleles, genotypes, strains, variants, transgenes, cross-references. I'll talk more about our gene descriptions in more detail later. And we have a lot of annotation level objects. We have diseases that are annotated to genes and genotypes and strains and alleles. We have phenotypes, expression, orthology. And orthology was a big one to harmonize on because that's really what brings us all together. So it was a lot of good work done there to make an orthology set that we can use across species. We have genetic interactions. We even have variant effects on our variants now, and 20 ontologies or more, plenty of more data to come. And so when I was sort of talking to Monica about what might be interesting to hear in this group is just sort of two main topic areas. How did we get there? How did we get here? How do we harmonize data across species? How do we produce interfaces that satisfy these really diverse user communities? And I'll try to talk about that with two examples. I'll talk about how we harmonized what we call affected genomic models, genotype, strains, populations. And then I'll give an example of how we are producing tools that can be used outside of the Alliance through this harmonization effort with the use case of our automated gene descriptions. And then the second half, I'll sort of dive in a little bit deeper about how we're trying to participate in the FAIR data paradigm. And I looked through some of the old presentations that you've heard in this group, and I know you're well aware of what FAIR is, so I won't go into detail about what that is, but just sort of how we've chosen to participate in that paradigm and how we're trying to help our users not only help ourselves ingest the data, but also help our users consume it in a robust way. So I thought I'd start with that harmonization topic by just talking a little bit about the general Alliance workflow, which really looks like it parallels some of the work you're doing, the workflow you're using in AgBioData, potentially. So we start sort of with a biological working group, and these are really our Alliance data specialists. They're folks that know a whole heck of a lot about whatever domain that we're tackling next. If it's about disease, we have the disease experts from all the different organizations. They're usually curators. They have some technical folks in those groups as well, just to talk about the technical details. And they do requirements generation for not only the ingest into the Alliance, but also view and search, the data download specifications. They're kind of the hub of each domain. And then down the pipeline a bit are these data wrangling groups, and we establish data exchange formats. We parse and load and validate the data. We work on integrating the data. We make sure that the biological working groups know about the different other biological working groups and how those data overlap. Of course, we have technical teams downstream as well, working on our API and our search interface, and of course, our user interface. And it looks kind of like a nice long linear diagram here, but of course it's not. Anyone who's done development knows that this is a very iterative process, and we go back and forth in little eddies between all these different domains, trying to figure out the best way to communicate our requirements to each individual group. And I think that's really a theme that I want to communicate, is that the communication between biology and technology is so critically important, and having really solid definitions of terms and using that nomenclature throughout our pipelines is really helpful in speeding up development and saving time downstream when you're actually, working to produce something that shows the biology to someone else besides the people that are actually working on it. And I went ahead and I asked some of our data specialists, because one of Monica's questions was, what are your lessons? What lessons have you learned with working on this? And I thought they gave me some really good quotes. One person said that oftentimes we're using the same term to mean different things, or different terms to mean the same things, and it happens all the time. And we know that harmonization has many parts. It's not just the shared data structure, but it's the shared display, and making the data available for consumption in bulk is really important. And I think we've sort of been lucky at the Alliance that we've all come together and figured out that we're ultimately or inevitably creating something new here. You know, changes are going to be made in the source institutions, and they're going to be made at the Alliance. But if we're doing it right, we're sort of making a resulting product that will be more reusable and robust than the components. So we had some goals when we were talking about any kind of harmonization. We want to first and foremost establish that shared nomenclature that we can use for objection relations as they move through our system. This helps both upstream and downstream consumers of our data. We obviously want to use existing definitions if we have them first. If we can all use the same ontologies even, that's great. I mean, that's the ultimate goal. If we can't, then we don't want to make the goal of harmonization make everyone identical. We can't do that. There are definitely, like I've said, there's different user communities that want different things out of our data, but it does mean making something that's manageable by all the parties involved. We can't make it too terribly hard for one group to do it and super simple for the other. And there's tools we can use to make that work. We can make ontological bridges that allow certain ontologies to reference the other ontologies in the database. You know, we can make cross-references and SLIMs and mapping files. And we have other tools that help us do that harmonization. So I think probably the easiest way to see our process, though, is to look at a couple of case studies. The first one I sort of want to go through is this disease annotation object a work we did. It's a good way to describe it. So I'll just launch into that. So when we were talking about ingesting disease and phenotype data into the Alliance, the first thing we had to really focus on was, what do these disease annotations, what are they in reference to? What object or what thing do people want to find when they're looking for diabetes? And this is sort of how we started. You know, yeast, SGD and RGD and WMBase had annotations of disease to genes themselves. And Flybase and RGD and WMBase also had annotations directly to allele. And then if you're looking sort of down into the vertebrate world, MGI and RGD and ZFIN had annotations to genotype strains, or at ZFIN we call them genotypes plus SQTRs, which is just genotypes plus morpholinos or CRISPRs or TALENs, because different things affect the disease annotation differently. So we kind of had a widespread here. We didn't have a whole lot of harmonization on even the object type that we were using to annotate disease. We didn't really have calculations that were consistent on the Alliance side that enabled us to sort of like show a genotype level disease annotation on a gene page. We didn't have enough information when we started to do that. We didn't really have clear rules on allowing us, from a software point of view, to combine objects where the technical attributes were similar enough to avoid sort of special casing each page to accommodate all of these different kinds of base level objects. And really our goal was on a result page, on a page that says diabetes on the top, you should be able to see all the annotations from all the mods in some sort of coherent way. And so we just started working on, we started peeling this onion back, and we established rules that allowed some of these higher level annotations to be pulled back into the gene or into the allele. And we started making that more harmonized. So we established rules that said, you know, at ZFIN, if the genotype contains only a single allele, and that allele has, you know, is a point mutation, has a variant that is a point mutation, and we can basically say that that's a variation of the gene. The gene is an alteration that gene is causing the phenotype. So we're calculating things and putting them back onto the gene page as we can. You can kind of start to see that there's some harmonization there. And then, you know, to get down, let me go back one. So to get these kind of compressed, we had to come up with a sort of an overarching word to describe all of these different concepts that are very similar, as you know. So we kind of went back to the biologists and talked to them, and they came up with this really great term called affected genomic model. And you don't see affected genomic model as a concept on our website much. You know, that's not a term that we want people to necessarily learn. We want them to see genotype and strain and fish and things that they're used to. But what affected genomic model did is it gave us sort of a tagline to allow technical teams to combine genotype, strains, and fish into one thing with similar attributes. And so as you can see, we're starting to harmonize, we're starting to pull things together. And now we only have 3 main objects that we have to work with. And most of the people in our consortium can apply disease annotations to each of those 3 objects. So we have sort of a consistent annotation space for them. So what are some languages learned? I think one of the biggest ones for this particular use case is that when you're harmonizing something, using consistent language throughout the project results in the software stack being easier to communicate. Sometimes you have to build something new, that affected genomic model, in order to support the use cases consistently. We tried to design for these use cases. We're not trying to make a spec that everyone has to adopt. Like I find it hard to imagine that someone is going to adopt affected genomic model with robustity and use that throughout their site. It's a word that helps us harmonize. And then, you know, figuring out a system for change management early is really important. So when we started with those 5 different bubbles, all different kinds of objects, we knew we were going to have to do this in steps. We knew we were going to have to take the first step of trying to get all the annotations on the gene and trying to get them all in the allele. And then we could take the larger step of trying to get the ones for genotypes and population. So we had to build a system that allowed us to do some change management. And so that's sort of the back end harmonization work that's going on. I think another really good example of harmonization at the Alliance is the automated gene descriptions working group deliverables that they did. And just to give you a little background on what I'm talking about, gene description is this like short human readable text summary of gene function. And you can see them at the top of Alliance pages. And they're called automated gene descriptions because they're regenerated every time the Alliance builds a new data set. Before the formation of the Alliance, mods had these. Some of them were manual. Some of them have moved to automated descriptions. And with the formation of the Alliance, it became really important to generate sort of standardized descriptions that would help in gene function comparison. So the main goal of this was really to generate these modular standardized gene descriptions for all of the Alliance species. And I just wanted to give you a little overview of how we do that. So we were sort of talking before about how we put disease annotations on a lot of different base level objects, while we also, you know, curate with a similar ontology in all of those cases, a disease ontology. So we take sort of the ontology terms that are associated with that gene. We have a template builder for these sentences. So you can see you're taking, I'm gonna mess up that word, so I'm not gonna say it. So I'm gonna say intestine. So down here somewhere would be, is expressed in intestine, and anatomy turn hypodermis. And you can see sort of with the terms and the sentence templates and our really fancy algorithm that we created, we come up with this nice text description in the result as the result. So what makes a good discrete gene description? You can read a lot more about this in the publication that Valerio Arnabaldi and Ranjana Kishmore from the Alliance wrote with a lot of others up there. But what they'd said is that really a gene description should be readable, should have a maximum number of terms for each data type. It's for humans, it should be manageable for them. It should be specific. It should have information, content, and granularity of the terms with respect to the respective ontology. You don't want just to have a bunch of gene descriptions that say biological process. You want them to be specific. And we want the gene description to be comprehensive. It should cover the entirety of the annotations on that gene. And so we did that. We developed an algorithm that gave a predefined maximum length, or you take a predefined maximum length of terms and generate a gene description that sort of maximizes or highlights these 3 needs. And the other thing that's kind of cool about it is that not only can you do them at the Alliance, but there's a robust software package and data distribution system at the Alliance, so you can go and retrieve those gene descriptions via the Alliance API or via our download files, and you can see them here on different model organism pages outside of the Alliance. ZFIN actually grew from 0 gene descriptions to 23,000 over the course of this project. And so they're widely available for consumption. Like I said, the code for this is open source and it's documented and it's species agnostic. So you could actually take the gene descriptions code from the Alliance and put it on any kind of organism, specifying the ontologies that you want to use, and it would generate this nice description for you. And then finally, like, you can pull these back through some of the data availability pipelines that we have, which I'll go into a little bit further in the next section of my talk. So I think that's been a real good success on harmonizing together and making tools that are available outside of the Alliance, sort of on organism and agnostic. So those were kind of two case studies about how we do back-end harmonization, how we do front-end harmonization, but how do we get the result of that harmonization out of the Alliance, and how do we get it into the Alliance? How do we make those really important efforts of bringing the biology together real in terms of software development? And that's where I'm going to talk about JSON schema and our use of that, a little bit about our file management system, our API download files, and our data dictionary. So I'm sure this group is well aware of how the way we deliver our harmonization matters. We've all worked with disparate data before. We know that their entire careers based on wrangling this text-based data. We know what it's like to have multi-value fields, objects whose properties span multiple lines in a flat file, documentation that when it exists is separate from the file itself or incorporated into a header whose lines have to be parsed differently than the rest of the lines in the file. We also know how hard it is to itemize metadata without those headers. We need them. And we know what it's like to have identifiers that aren't unique or become obsolete without any support for how to replace them with new IDs. And we obviously know what it's like to find integers where we are expecting strings and vice versa. Finally, we've probably all written custom validators or parsers for files that require a lot of upkeep and maintenance. And so this is just my slide of trying to show you how at the Alliance, even as a group, we are interested in this as well. We have to do the parsing, we have to comprehend, and we have to compare all this data together. And so how do we get there? In a perfect world, the data specifications we used at the Alliance really should be self-documenting. We don't want to get into the problem of it was too long, I didn't read it. You know, we want to be self-documenting. We want to have change management built in. We want to write code and data specifications that don't make a downstream user change their entire parser just because we want to add a field or change the logic of it. And we want people's parsers to be limited to supporting their business logic for their organization and their local data store, not handling multi-valued fields, doing type checking, unexpected null values, all with the expectation that that's going to be cheaper and easier for people to use our data. And we want to enable generic validation methods that don't have to change based on the changes in the file content. We really want to be able to write a validator that works on all the Alliance data, not specific files. So in all of that, we're trying to standardize our inputs to reduce the cost of exception handling for each species downstream in our software. So in a perfect world, we would choose something like JSON schema, and that's what Alliance did for a lot of our ingest, right? So I'm just giving you here sort of an example. You can go to this website here and see, or the GitHub repository for it, and see all of the different ingest schemas for the Alliance. I just wanted to highlight a few things here about why it's nice. I'm sure you've all worked with JSON schema too, so you probably know this, just stop me. But all the fields in this specification are typed. We know whether it's a string or an int or a Boolean. The properties are defined as required or optional. You can get a sense of how these objects are related to each other based on, just based on the content of the file. They have the ability to have these reference points. If you can see, I'm not sure if you can see my mouse, but in the line 11 there, it says ref, and that is actually is referencing a whole nother object in the file. So let me just pull up that whole other object here. And that whole other object has its own set of attributes and values that are possible. And so we can really create a modular schema. This screen that came up just now on the right that says basic genetic entity is actually reused through several different of our ingest specifications. So we can take advantage of reuse. We see when something's an array, and we see what kind of values to expect in that array right from the specification. And I just wanted to show this really quick because it's kind of cool. So for all of that 40 different data types that we have or more in the Alliance right now, bunch of annotation types, all of it's validated coming into the Alliance with 45 lines of code. the best part of it is this validation hasn't changed in years now. Chris Tabone at Alliance wrote this 45 lines of code right when we were starting, and it's been used ever since to make sure that what we're getting in matches what we're expecting in the schema. And so it gives a wide variety of tools to the data modeler that require very, very little code to maintain. So they can do all the fancy specification in the JSON schema, and we can validate it and resolve it with just 45 lines of code. So of course, like, we can't always use JSON schema. It's a relatively new, well, I probably shouldn't say that, it's not that new, but in terms of our domain, it's sort of a new technology that we're working with. It has its downfalls, obviously. Like, if you don't really know how to participate in the JSON world, it's very difficult to get wrap your mind around a file like that. It's not just a flat file that you can, look at in Excel very easily. So you kind of have to, that's a downside for sure. And the second downside is that there's existing standards out there that have been used forever in our domains. GFF3 files, VCF files, they're like the backbone of a lot of our work. And even at the Alliance, we have to be able to process and use those file formats as well. So I think the stance we've sort of taken at the Alliance is that we do spend time making a specification for GFF that is specific to how we want to work with the data at the Alliance. And I know that Monica and Meg have gone through this as well. And it's been very, very lovely to sit on those conversations with you all, because you're facing the same sort of issues that we are. And it's nice to have the idea that we could have one spec sort of to unify some of this data. It's really kind of cool. So this is just a slide that shows you our work in that regard. I mean, we have variants and transcripts and exons and CDS records coming in through GFF. We have VCF files. We use a tool called the Variant Effect Predictor, which is a tool out of EBI, and it kind of runs the variants through a pipeline and tells us what their consequences are. And then we slurp in those consequences back into the Alliance. And it was really important, in order to get these three tools to sort of function together, that we had stable identifiers in our GFF3 file, that those stable identifiers had consistent naming and consistent ID resolving ability, and that they were in the same column in column 9 of the GFF3 file. We're still working on that. And then in the VEP, we had to make sure we understood how they were using those GFF3 files to pull the data back. So long story short, we also tried really hard to work with existing standards whenever we can. And if you want to see sort of our GFF3 spec and what it looks like in the Alliance, I sort of linked it down there. It's not formal yet. We're still in the process of doing it, but it does give you an idea of where we are with that. So we did all this work. Is it even reusable? We think it is. So I wanted to show you a little bit about how we've seen reuse already happen with our Alliance workbase, and then give you another case study of how we used these JSON files for a particular user, an actual user, not just all of us Alliance folks. So first, before I do that though, I wanted to tell you that one of our main focuses in all those JSON schemas and ingest pipelines was that we wanted to make it possible for other species to jump in and start submitting data to the Alliance in a known format. And we kind of just tested our theories of whether or not we were doing this correctly by trying to ingest the SARS-CoV-2 genes and molecular interactions data for SARS into the Alliance. And what we found is, if we did format the data, there was work that we had to do to format the data into those JSON specs and ingest files. But once we did, the data moved fairly well, fairly easily through our system. I think the biggest thing we kind of ran into was that virus genes and viruses are much different in terms of display on a browser, like a JBrowse instance, than the rest of the genes. So I know Scott Kane did a lot of work, and his team did a lot of work to make that work in JBrowse. But in terms of moving the data into the Alliance via our gene ingest, it went pretty quickly. We have a genome widget on the Alliance site that's a standalone widget that shows the browser inline in a gene page. And I know that that's being reused outside of the Alliance. Of course, we have JBrowse, which is the ultimate reuse of all this data. We have gene descriptions. I talked about those a little bit. And if you look on the Alliance site, there's a summary ribbon. It has a lot of pretty squares in a line. And that ribbon code is used not only at the Alliance, but at ZFIN and Flybase and other places. So we are doing a lot of reusable work here as a result of the harmonization. One project that's coming up that probably has spoken to maybe some people on this call, I'm not sure, is we are making an Intermine instance for Alliance, and that should have the normal Intermine hooks to all the other Intermine instances in the world. So that's another place where you could do it. But let me get into this expression use case study that we did with zebrafish data. So we had a group come to ZFIN and ask us for expression data. They were looking to verify some research algorithms they were working on. They were looking at RNA-seq data, and they wanted to verify that expression data sets at ZFIN sort of validated what they were finding from RNA-seq. And I don't know all the biology behind it. I just know about the data. So bear with me. That's kind of the summary, though, of what they wanted to do. And of course, ZFIN provides expression files, and we provide them in standard text formats with column headers. Oftentimes there's a back and forth with ZFIN staff to sort of identify which download files are needed for a particular user. We talk to them about how to interpret the column headers, how to combine the files as necessary to get the data they need. Usually these files are pretty generic, you know, they need to get a handle on how to parse them, etc. And so we were given the, you know, it's kind of a fortunate step that we had a couple of users that were willing to do an experiment with us. So we gave them the Alliance ingest specifications for expression data, and we gave them the files that ZFIN is submitting to the Alliance to see how they could use those. So I just wanted to give you an example of what we gave them. Here on the left is that JSON schema that I was talking about. You can see each of the attributes is documented in line, has references that you can navigate in that GitHub repository. And then the file they got, this kind of lovely JSON file. You can see at the top here, we have our header integrated into the file itself. So it's parsable just like the rest of the file. And you can match up the attributes here. Oops, I'm sorry. Oh no. You can match up the attributes here, cross-reference, you know, you can see it right here, cross-reference, what does that mean? And when expressed is an object, you can see that over here, when expressed, and you'd have to go to another screenshot of the when expressed schema to see what you're finding there. So we gave them all this, and the results were that the users were really able to use the specifications quite easily. There was less back and forth. They automated their data to ingest, they parsed and loaded quite quickly. But there were some caveats to that. One of the things we found with the users that tried to use it were that if I can just go back a slide, you know, when we give someone an ontology term, sometimes that's not straightforward to them on how to retrieve the actual name of that ontology term. You know, processing an ontology file is another step for them. So they really wanted us to just give us the human readable ontology term as well. They needed help translating those into names. They wanted fields like gene symbol instead of just the identifier. So what we found though was that we were able to take the Alliance ingest spec and add or extend the JSON schema a little bit for ZFIN for this user. And that expansion didn't necessarily break any existing parsers that might have been using the spec for Alliance, but it did allow us to expand it very quickly. So we took that as a, you know, at least 70% good result of this experiment. And we took that further into the Alliance and said, well, okay, we're definitely going to provide TSV downloads of all of our data. We definitely need that. You can go here and sort of see that. We also provide a JSON version of them as well. And of course, we have a API at the Alliance that also allows users to retrieve our data via these API endpoints, and it all comes back as JSON. If you want to explore that a little bit more, you can kind of go to the genes link here that I provided, and we're always looking for feedback on how people use the data. So if you have any feedback, or you've gone and you really want genes, you know, and you've tried to do this, we'd love to have your feedback on how it's working. That's probably the most important part. And this seems a little bit out of context of what we've been talking about up to this point, but I really wanted to put a couple of slides up to talk about our file management system. You can kind of go see the code there in that link, and you can see the output of the what we call the FMS, the file management system, in this API call. And this is our system of tracking these ingest files, and also it also tracks our output files. So it tracks all those download files that I showed earlier. It tracks all the ingest files, and Olin Blodgett at the Alliance did this work for us. And it provides not only an ingest API. So let me step back. So it gives us an API that we can push our data to, that validates the ingest on ingest. So it runs through that JSON schema validation, and it tells us in real time whether or not our file passed the JSON validation. And then it can also, do validation for other non-JSON files. Like we're working on a GFF3 validator that would be triggered by an update to the file management system as well. And then it also provides a download API. So the code that ingests these files into the Alliance goes again to the FMS, and it hits this endpoint, and it pulls down the files for a particular Alliance release. So let me just show you a little bit what that looks like. Here's our API endpoint response for the FMS for the latest release that's on production right now for Alliance 3.1.1. Oh, where did it go? Oh no, there we go. And so you can see that the data files have a path to download, they have an MD5 sum, they have a data type, and they have an upload date. And this provenance is tracked through all of the different data files in the Alliance. So it's a very nice system that we use quite frequently for everything we do there. And sort of the last part I want to talk about is our data dictionary. And I don't know if data dictionary is a term that is familiar in our industries yet. You can kind of go look at our data dictionary here in the GitHub repository, but a data dictionary to us is a little bit different than the JSON ingest schema. We get a lot of our model from the JSON ingest schema, but we also have to do work on the Alliance side to process the input into a model that, at the end is what the software downstream uses. And what we noticed in the course of this project is that, of course, the flexible nature of our data can result in data inconsistencies that aren't necessarily caught on that nice JSON validation that we have. And we know that biological models have to be flexible. What we really want to prevent, though, is unexpected data inconsistency. So we know data is going to be inconsistent, but if it's unexpectedly inconsistent, it really increases the cost of software maintenance. So a few examples of this that we went through at the Alliance, a couple of years ago now, but you can kind of get the point. So when we're ingesting genes, we noticed that the sequence start and end positions that we processed from those JSON JSON files were stored as a result of our Neo4j importer, not of the JSON schema, but of the Neo4j importer, was stored as text instead of integers. Sometimes we would do cross-reference processing where the URL would get missed. It's not something you could necessarily, we don't want people to submit those URLs. We want to generate them for them, so the generator would break. We'd have sometimes inconsistent ontology loading. Some ontologies would have part of relations, others did not. We didn't have in Neo4j a way to enforce the existence of attributes or relationships except through tests. So we wanted to come up with a system where we could be more generic. And the data dictionary concept is what we're currently working towards making a reality. So let me just show you some examples, because that was a lot of words. So this is what a cross-reference data dictionary at the Alliance looks like. It's YAML, and we chose YAML because the BioLink, I'm not sure if you've heard of that, is written in YAML. Other data dictionaries are written in YAML. It seems pretty easy to read. It's got less punctuation, mostly for readability. And so you can see, let me just start advancing the slide a little bit. So a description can be a biological definition of our object. Not null can be used to validate whether or not that cross-reference URL should be null at the end of all of our processing of our data. We can use properties to define what downstream software users can expect to find on a node. And it's important when, they can, in our sort of flexible graph store, all the nodes can be differently decorated. So we really want to try to have a contract that we maintain between those different nodes. And then of course it has a relation section that determines the way two nodes are related to each other. A cross-reference has a many-to-one relationship with a gene, for example. And so what we can do with this data dictionary specification, or is we can take it and parse it and use the definitions to validate each and every node and relationship in our database. And that's a lot easier than writing tests for every single use case, every single relation and object. We're still working on this. It's not a reality yet, but that's the theory. So again, it gave transparency to the Alliance data model. More people can contribute to the shared understanding of those nodes and edges. I mean, it's one thing to have biologists robustly defining these JSON schemas that get input into the model, but then they also need to have input on the resulting database that gets generated from those ingest files. We think it encourages a shared language throughout the software hierarchy. We definitely don't make that data dictionary in a vacuum. We have to take into account what the user interface is thinking and what the API needs to do. And it provides a mechanism of a sort of a readable format to validate those nodes, reduces the need for handwritten validation tests, and we think it could be distributed as a documentation tool, although I don't have any use cases for you yet, so we're still in the process of doing that. So just to come, it's more of a future to come work we're doing. So just a summary, I wanted to give a sort of a timeline of what we've been doing. We launched in May of 2016. It's now just a little bit after May of 2020. We touched on a lot of these different milestones in the talk today. We started our AGR schemas, our JSON validation, ingest validation right at the beginning. We started storing all of our files on S3. We quickly noticed we needed that file management system to do provenance tracking and data ingest and export. We started our API. These are kind of the technical milestones that we've went through. And then down at the bottom, you can see sort of the evolution of how we added data. And so we started at genes orthology and ended up, now we're just working on high throughput expression metadata, more work with variants. I highlighted again here the two times we looked at GFF3 files to see how we could interoperate and support the data that we needed. So gist of this slide is basically we're still very much in progress. And it's been, like I said earlier, it's been lovely to hear other people's experience with this kind of work. I think there's a lot of work we can do in common here. And it would be nice to work together as much as we can to reconcile this across not only animal species, but plant species as well. And this presentation is the result of 150 people's work. This was our last picture we took together at our face-to-face meeting. It's not all of us. If you want to see all of us, there's a big wall of text for you to see. I'm open to questions, obviously. I just wanted to point out before I open it up, in case there are questions, to a couple of slides at the end of this presentation that show all of those links that I have throughout the presentation, and 2 plugs from some of the people at the Alliance that knew about this presentation and wanted to sort of open the door a little bit for more participation together. Sue Bello invited everyone here who's interested in phenotype ontology reconciliation to the Phenotype Editors Call. I'm sure some of you are already on that. There's some details there. The Alliance folks would welcome any kind of discussion at the PI level about shared interests. And we have a user forum email list that's coming out soon. I don't have the link to it yet. We're just in the process of developing it, but we do have a case in to at least bug Monica to see if she would like to sign up for that, the user forum, to discuss what we can do next to harmonize. So that being said, I think that's all I have. Is there any questions?
Yes, so this was fantastic, Sierra. So, but I hear there's a question already. So
yeah, a request. Could you go back to the slide with the links on it?
Oh yes, let me see.
And maybe have that set up while you.
Okay. Yes.
We're all frantically looking at those links now.
That's good. That's good. Let's hope they're working. No, I'm just kidding.
I'm kidding. Well, maybe I'll go ahead, Sierra. This was really great to hear how these well-established groups are surmounting these data harmonization challenges. I had a bit of my GFF3 hat on while listening to your talk. And for those of you in AgBioData who don't know, we also have currently an AgBioData GFF3 working group where we're working not just within AgBioData, but across with Sierra and other people from the Alliance and CBI and other commercial software groups too, to see if we can make a better GFF3 spec that is less breakable. And this was a working group that was spearheaded by one of Maggie Woodhouse's questions and ideas. So and Sierra is also on that working group. So I was looking at the fact that you guys use JSON instead of GFF to exchange data, and what you said made a lot of sense. Do you think, dare I say it, GFF3 should be obsoleted in place of something else like JSON, or do you think that there's still a value for it somewhere? Sorry, big question.
That's the best question ever. I think what it is that GFF3 is just used so consistently in so many tools that it's really hard to just, throw in the towel. And as you said before too, it's really difficult to do that. I know that there are some object-oriented ingest specifications for GFF3. Of course, I'm blanking on it right now, but Chris Mungle spearheaded some of that work. And so there is an opportunity for us, I think, to investigate that. I know that people actually using GFF3 would be, I bet Scott is probably has a lot to say too, so I'll let him answer as well. But I think it's not a bad idea to look at that, to see what it would mean to produce a different file format than GFF3, with the caveat that it's so widely used and it's in so many tools that are we just shooting ourselves in the foot? Because then all those tools would have to change. It's a really hard question to answer. Scott, do you want to say stuff too?
Yeah, well, I'll just chime in. First of all, I have to note it's funny that you used the word consistently when you were talking about the fact that it's used in a lot of places, because yes, that was a perfectly fine word to use in that instance for one word of consistently, because it's ubiquitous almost. However, the thing that is funny about it is that there's one thing we know about GFF3 is that it's rarely consistent when you go from one to another. But I think the main point that you made is probably spot on, is that there are so many tools that generate or consume GFF3 that kind of deprecating it might be really, really hard, or at least a really long process. I think that I could certainly imagine creating, say, a JSON spec that is more, it's easier to validate, because JSON is easy to validate, like Sierra already showed. So I could imagine creating a JSON spec that tries to do everything that GFF3 does, but does it more consistently and better. And then all you need, in quotes, then all you need are tools that will either generate that JSON or, basically take GFF and generate the JSON, or take that JSON and generate GFF. I think it would be a big project. I mean, I'm talking. And the other problem with it being a big project is it would be a very distributed project. It's not like you could hand some group like me a big pile of money and say, get rid of GFF3 and make it happen, because I don't think that's the way it would work. Unless, I don't know, give me a big pile of money, I'll set up a consortium that will hand out money, smaller pots of money, while taking my massive cut. Then maybe it would happen. But I think it would be a really big project.
Point taken, yes. And I think one of the things that we've run into in discussing GFF3 issues in our working group is that, there's the format, so it's a 9-column structure, but how those different fields are interpreted is part of the issue. And so I was really keen in looking at your description of the data dictionary, Sierra, and your work there, and wondering if that might be one way for us to communicate what we think some of those fields should mean. But I guess we'll see how things go with you guys as you develop the data dictionary and see that.
That's actually a really good idea. That's a great idea. Yeah.
Yeah, because we were thinking, like, should we stuff all that information in the header again? And maybe we just need a YAML file or something to have a machine readable way of expressing that. But I'll stop talking about GFF3. Are there any other questions for Sierra?
So this is Doreen Ware. I'm from USDA. I posted it in the chat. I was curious, what is the mode of how you guys actually work together as a group? How frequently do you meet? You know, how many working groups do you have? Just to give us a flavor.
I think Ann Eagle might be on the call. I saw her in the chat a little bit. Ann, do you want to address that, or I can go over it too if you'd rather.
Sure. There's a couple PIs here too who might want to chime in, but the Alliance started 4 or 5 years ago, and we started out with the paradigm that we would have biological working groups that would come together, harmonize data, and then create requirements, and developers would be part of that as well, to then take the harmonized data or the specs, the JSON, load it, and then display it. Boy, that sounds so easy. So we started out with the disease working group, disease and phenotype and expression, and genome features, which included JBrowse and a sequence viewer that we show, and basic gene information was another one, and gene descriptions were, so I think we started out with 4 or 5 core working groups, and to stand up a site. So that is now stood up. We've had a site for we're over 3 years now. And so many of those working groups are still existent. Those working groups consist of at least 1 member from every mod and go, so that there's representation for all of the data. And that's on the biological side. On the development side, developers were volunteered or assigned to work on the Alliance, and we came together and figured out what technologies we knew, what technologies we felt would be appropriate for a new mod, in essence, gave us an opportunity to try some new things. And so it kind of organically developed what our software stack would be. And that has continued over time. And now we're starting to think very much more long-term. How do we do curation at the Alliance? How do we pull publications? How do we store publications? So we're starting to think much more long-term than because now the basic data is on the site and available. Does that make sense? So I don't know, we've probably got 12 or 15 working groups at this point, looking at different parts of the data. And those working groups can all be found on the alliancegenome.org site. There is a list of them and what their specific tasks are and what their deliverables are.
So, Anna, go ahead.
Okay, I'll just go ahead. There's like 5 of us trying to talk. So this is Sabrina. I'm also at the Alliance. I am a curator. So one thing I wanted to add to that is that depending on what we want to add at the Alliance, so this is a decision that is made at the PI level, then the working groups, the biological working group put together, and each group meets, most of them meets once a week for about an hour. And some working group meets a little bit more often. So maybe we would have extra meetings when we have a deadline coming up and we need to have more discussions. Some working groups meet less often, so every other week or once a month, depending on what needs to happen and how what the priorities are and what needs to be discussed, obviously. So we have a big range of, you know, how often do we meet? But I have to say this, when you look at the calendar for Alliance, we meet a lot.
This is Chris Grove from the Alliance and Wormbase. I just wanted to add that I think Sue Bella already, she put a link in the chat, and I think Ann Quiddick also just put one in there. There's a link to the working groups page. There's currently 18 lists that have been instantiated. 15 of those are currently active.
and this is Jennifer from RGD and the Alliance. And just to kind of give you a little bit of information about the active versus maintenance mode. The idea that we had at the very beginning was any particular working group would be spun up, would meet for as long as was needed to get that particular kind of information into the, or that particular kind of data into the Alliance. And then for most of them, the idea was that then they, once that was in, then they could go into a maintenance mode where if there were problems, the group members of the group were still available, but they wouldn't meet on a regular basis. So there are a number of groups that are in maintenance mode. And there are a few groups like the disease group and the variant group that there really is no end in sight at this point. There's just too much to do, and too many things to talk about to really think about spinning those groups down into maintenance mode. Those will probably be active for the foreseeable future.
I think this is Doreen following up. Punkage has a question. Is there any possibility for the people in the plant group to participate in those working groups?
I was sort of quiet, hoping that one of the PIs on the call would talk about that. I think when I asked that question, I knew it was going to come up, and I think that it's so important that we have these models and these things exercised by more than just the people in the groups. That's just critical. And I think what their kind of roadmap with that was that maybe some people from this group should come and talk to the Alliance PIs and figure out how we can make that happen. And I think all of the working groups would welcome participation, but we got to kind of figure out how to channel that through the appropriate people first. Does anyone else want to say anything to that regard from the Alliance?
Yeah, I think that's fair. It would be great if you had us come and speak to you. It would be great if somebody from your group could maybe come and sort of talk to the PIs at the Alliance, and just to get a sense of what are your goals and what are you looking for, and how could we potentially harmonize plants and animals, and as far as the observers, I think that is, I think we have some observers from other groups on some of the working calls, but I think it would be good to just kind of chat about that at the PI level and have somebody come. So Doreen, you asked the question, do you have somebody that you would nominate to maybe represent and come and speak to us?
I think I could talk with the rest of the AgBio group and come up with some recommendations for that. And it may even be possible for us to even target, depending upon which of the working groups, what that might look like. But I definitely would be interested in following up on a call with you.
That'd be great.
I agree. So we'll coordinate internally on how to handle that. And there was the question of how to share data between plants and animals, but also between plants is what we're also talking about too, and within agricultural data. I see another question here. Who all from the plant community is on the Phenotype editor call, from Tanya?
Okay, so this is Sue Bello, and I'll have to go look that up. But just to note that the Phenotype editor call is actually independent of the Alliance. So there are a number of Alliance members who go to that call, but there are also a lot of non-Alliance mods that are in that same call. So that call is actually, everybody's welcome to join if they're interested. I think it's somebody from the plant trait ontology who's usually there. I can go look up and I'll let you know.
This is Doreen. I also have another question I didn't type in, but I'm curious, how closely do you guys in your Alliance coordinate with the Alliance for Human Health, the Global Alliance for Human Health Initiative? Or are there any overlaps with that?
I don't think we have any direct overlaps.
Okay.
No, maybe members of the Alliance are also involved, but I don't think there's any coordinated overlap with that. Is that fair to say, Sierra?
Yes, thank you for answering.
I will just say that on some of the things where we've defined things that have also been defined by the GA4GH, we've reviewed, and I know Carol Bolt from the Alliance was going to take some of our questions and issues back to them. So we do try to have a back and forth conversation.
You too.
All right, thank you for the questions. We are over our time. So I think at this point we should thank Sierra for joining us today and giving us such an excellent presentation and food for thought. And we'll certainly, within the data sharing working group at AgBioData, but also beyond, we'll need to think about how to take some of the lessons that you showed us and learn from that and help inform our goals within AgBioData for data sharing as well. But it was really great to see, wow, what an excellent job you all have been doing for that. So keep up the communication. Are there any more questions? I think you all have Sierra's information, or I can help pass your questions along. And have a great day. Bye, everyone.
Thank you. Bye.
Thank you.
Thanks.
Bye.
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 Bye. Bye. Bye. Thank you.